Madam Chair, ladies and gentlemen. When Angelina Jolie told the world that she'd had a DNA test and had decided to have her breasts removed, we were all shocked. But I suspect very few people asked, what's a DNA test? Everyone knew about DNA, thanks, of course, to two famous people, O.J. Simpson and Monica Lewinsky. It's easy to forget that before that popularisation, this was very much the reserve of the medical elite and academics. And if we go back to 60 years ago, there was very little penetration of that iconic discovery. Indeed, 60 years ago, my mother was mainly distressed by the fact that our newly bought television didn't work when she was trying to watch the coronation, because the lady next door had an unsuppressed vacuum cleaner, we later discovered. And I was deeply distressed by the fact that my mother had withdrawn breast milk, and I, in fact, apparently spent several months subsequently trying to gain access to her bra without success. A behaviour pattern which re-emerged about 15 years later with equal lack of success for many years. This is a great honour and pleasure to be here back in UNESCO. This is my third visit to this room, and in fact this time last year I was sitting here, not falling asleep but reading that monitor, when Professor Lee, who is here in the front row, was presenting the endorsement of the Human Variome project by the Chinese government, a very important step, and pledging their support to this really very important exercise. And what I'm going to do in the next 20 minutes, with your permission, is to speak a little bit about the Human Variome project and why I think it is an important project, one that we should all endorse and support, despite the challenge that it offers us. And I'll use 3 or 4 clinical examples, which for the experts in the audience are very familiar, but I hope you will forgive me using them. I actually became a geneticist, I think, 45 years ago. I was a biology student, rather bored, got taken to a lecture by a geneticist who explained the genetic code. What captivated me was its simplicity, and the lecturer told me a little bit more about it over coffee at the end, but what really captivated me was that my biology teacher didn't understand it and asked me to explain it to him on the way back to school on the bus. And that memory stuck with me. And when I went to medicine and discovered that there was no genetics in medicine, I decided to take a year out and study it and acquire a degree in genetics, and that set the course of my career. What I learned back then, 45 years ago, was that in fact it is a degenerate code, and it is in fact very much that which provides the basis for our challenge, because in fact if you take your 4 letters and combine them in sets of 3, when looking at the RNA rather than the DNA, you get a U instead of a T. The situation is you can actually create 64 different combinations of those 4 letters in sets of 3, but we only need 20 of them and a few full stops. So basically, if you take, for example, the code CCG, then you get proline. But in fact, if you take CC anything, you get proline. And so we have a problem immediately that when we look at the sequence of the DNA, we could get lots of different codes for the same amino acid. What was really quite striking as we began to understand genetics was that really quite simple changes could have a profound effect. This little girl from West Africa has sickle cell disease, and she has a swollen forehead because her bone marrow has hugely expanded to try and replace the constant breakdown of her blood. These are her red cells, not the usual donut-shaped, but they've gone a sickle shape because the hemoglobin molecules are all sticking together, bursting the cells, causing extreme pain, loss of circulation, anemia, and generally a very early death unless given intensive treatment. It's an interesting example of what we now realize to be a common issue, that in fact we are evolved, our genes have been selected, and this particular gene proves to be a great advantage if you are a carrier of it. If you inherit it from one parent, your chance of dying of cerebral malaria is dramatically reduced, as a result of which, in the Rift Valley area, up to 1 in 3 people carry this gene. Obviously, when they marry and have a child who gets 2 copies, that child manifests this disease and probably dies. But that is offset by the number of them who don't die of malaria, which I recently discovered has killed probably 10% of all humans who ever lived. And so that selective force has driven up the frequency of this gene. Back when I was studying genetics and before Professor Arber and his colleagues had solved our problems for us in terms of analysing DNA, we'd already worked out what the spelling mistake was in this gene by simple deduction from the protein. Clearly what had happened was there'd been a single letter change that had changed the glutamine to a valine, a glutamic acid to a valine, a very simple change which meant that in low oxygen tension your hemoglobin molecules stick together, causing this dreadful disease. It's really worth remembering, even for those of us who do this for a living, how our entire life can be devastated by a single letter change out of those billions of letters in our genome. And you realize immediately the challenge that we face in finding those single letter changes that can be so life transforming, and how we need to pool our knowledge in order to be able to achieve that. So sequencing began with Sanger, who invented this very simple, rather elaborate challenge in the old days of sequencing DNA, a chain terminating technique, the Sanger method, still generally the workhorse of our technology. In the old days, we used X-ray plates, which meant we were all carrying around big sheets of X-ray film to see where the fragment stopped in each of those 4 columns to tell you where the A's, C's, G's and T's were. Later that was given a fluorescent tag, and then of course the ABI sequencers came along which could read those fluorescent tags simultaneously. And so the Human Genome Project, of which we're all familiar, had serried ranks of these machines, and if you went to the Sanger Centre where about a quarter of this work was done, there was a huge factory, hundreds of people generating sequence using these machines. That same army of people could now be replicated by one technician with one machine. That means that we can now sequence DNA at a scale that even the most imaginative of us, I think, could not have foreseen a decade back. Craig Venter and Jim Watson both had their genomes sequenced in the mid-2000s. Followed the third, in fact, person to be sequenced was a colleague, a clinical geneticist in the Leiden Centre, Mylene Creek, who volunteered to have her genome sequenced by Johan Den Dunnen, who is one of our key leaders in the Human Variome project. And that cost about 40,000 euros. Had it been done in 2000, it would have cost about 100 million euros. So that was the profound collapse in cost. Interestingly, our American colleagues were deeply unimpressed by this achievement, pointing out that it had already been done twice before. The Australian media captured the moment, however, when they said that man takes one step towards understanding woman. Having been married 40 years, I can tell you it's a very small step indeed. So what we're actually seeing there was the first beginnings of a true revolution in our laboratory technologies. Gordon Moore described Moore's law for computing, how every, was it 6 months or 2 years, rather, our computers would double in their transistor power and density. And sequencing costs were falling much in line with that Moore's law for computing until the advent of next generation sequencing, when in 2007 the price just collapsed. And we're now down, I'm going to have my whole genome sequenced as a training exercise in September, and it will cost about £5,000. And it's confidently predicted that just using our existing technologies and some newer ones coming down the track, like Ion Torrent and Oxford Nanopore, we can probably get that down to about £1,000. And I'm working in a small biotech company who is competing with the world to think of an even better way of doing it, using nanowires. We call it last generation sequencing, modestly, and we think we can actually sequence chromosomes without breaking them. And so people genuinely believe that one of these technologies will take us to giving us the whole genome for maybe $100. Now, of course, it isn't a genome we can use in the clinic because it's got spelling mistakes, its density of coverage isn't certain, but nevertheless it demonstrates the enormous expansion of our capacity. So the first of the examples that I want to draw to your attention, I was sitting in the early hours of this morning thinking I'd like to put a couple of clinical examples in, and this is one that I quite like because back in the 17th century, Gerrit Jans and his 13-year-old wife, it was legal then, arrived in South Africa in Pretoria and set up home quite near there and started having children. And I was there only 10 days ago, so this one was stuck in my mind. And what was interesting was that in the 1960s, a Dr. Dean was looking after a nurse in South Africa who died unexpectedly when he gave her a barbiturate anaesthetic. And she had this unusual problem, what's called porphyria variegata. Porphyrins are the things you use in making the heme in your hemoglobin. And people with porphyrias can't metabolize those porphyrins correctly, and they become light sensitive. Their skin burns in the sun. And so it turned out that this nurse who died had that condition. And then another member of this same extended family died as well. And Dr. Dean wrote an iconic MD where he studied this huge pedigree. It turned out that 10,000 people descended from that couple had inherited that spelling change, which gave them this initially fairly trivial condition of burning skin when you get in the sunlight, until they were exposed to the new challenge of barbiturate anesthetics, when their much more life-threatening pharmacogenetic problem emerged. Now, it's a good example because this is a classic founder effect. You don't need to inherit this from both parents, just from one parent. That will give you the chemical toxins in your skin, the abnormal porphyrins that lead to the problem. And this so-called autosomal dominant trait has become very common in South Africa. And we see this all over the world. So clearly, when we begin to understand the human variome in all its dimensions, we can't simply sit in one place and look at the people who live in our street. This has to be a global effort in order to fully understand this variation. And of course, most of that variation is in Africa, because we left collectively quite recently, and so we're all quite closely related. And in some areas in Central Africa, the difference between villages is greater than the difference between the Chinese and the English. And so that's a hugely important part of this exercise. So this is Jim Watson. Jim is a friend and a patron of our Centre for Life in Newcastle, where we, as you heard, are involved in genetic education. And in fact, we have 40,000 school children going through our institute doing practical genetics, and 250,000 people coming to our visitor centre to see the science exhibits. And in fact, this September, one of the children who went through our centre has completed her PhD in genetics and is about to start in medicine and wants to be a clinical geneticist, so I have finally replaced myself. Jim came along with Francis Crick. Apparently we're the only project they ever co-sponsored to be our patrons, and this is Jim outside our department a few years ago. Interestingly, I had his whole genome sequenced and is perfectly fit. He has a habit of saying strange things to journalists, but we don't think it's genetic. We can't actually find the explanation for it. This, however, is his genome, and the team in Baylor assembled it, and it illustrates the challenge that we face. The missense changes, these are simple changes of amino acids, like the one I showed you in sickle cell. 8,967 of them. And then 2,421 we'd never heard of before. And the list goes on, spelling nonsense that stop the gene, etc. Synonymous changes that just don't apparently change the amino acid, but might change the way the gene works, and so on. And then when you get out into the areas around the gene, you find thousands more of these spelling variations. It's estimated we have about 3 million each, of which perhaps 400 are of major relevance to our health. And 1 or 2, like sickle cell trait, if you get them from both parents, could give you a dreadful disease. So we've got to sort this out, and that's where the Human Variome Project comes in. Returning to Holland, if you go to Amsterdam station now, you see that we have a problem. We have a traffic jam with bicycles in Holland. That's nothing, however, if you go to Professor Lee's hometown and look at the traffic jams in China. China currently holds the record for the longest traffic jam, I understand, although I think Moscow is trying to challenge. But this was in 2010. They had a 100-kilometre-long 11-day gridlock on one road. They had to set up a whole sort of series of villages to feed the people who were stuck on this road. This might look impressive, but it is nothing compared to the gridlock we are heading into now, as people like Joris there churn out vast numbers of exomes and genomes into our clinical databases. We have got to pool our resources to solve this problem. My second or third example is a condition in which I've had a long-standing interest to illustrate the practicalities of the Human Variome Project. This is a family with hereditary cancer that I was working on about 20 years ago when I was approached by a yeast geneticist, or we were approached by a yeast geneticist, to ask if he could have some DNA. He had an idea what might be going on, because families like this, their tumours pick up extra spelling mistakes. And Richard Culloden's team actually took DNA from Ted, a local shepherd, who had had 3 colon cancers and 9 skin cancers, and his sister Bertha, who had endometrial cancer, and demonstrated that they had a spelling mistake in the gene that corrects spelling mistakes, the mismatch repair genes. It transpires that yeast and indeed E. coli use essentially the same genes to repair their DNA, so that by taking the DNA from Ted, he was able to actually identify, using the yeast equivalent of those genes, his copy of those genes, and find the spelling mistake that caused his cancer. It's an interesting thing to reflect on philosophically for a moment, that in fact, when you're sitting here, or if you're sitting in a pub drinking a glass of beer, there's a yeast cell somewhere down in the glass actually fixing its DNA with the same genes that you fix yours. If you've got no one else to talk to, you can reflect on that. It's a sort of a Buddhist moment. The importance of this was that this gave us an access to a whole new class of disorders, that we could actually start to predict which members of these families would get cancer and which would not, and actually intervene. And indeed, with Professor Finn McCrane, who's in the front row, and colleagues around the world, we've recently demonstrated that simply giving aspirin to these patients can halve their cancer rate. And we're about to start a new trial to see whether we can achieve that with low-dose aspirin. So this is really very important, to be able to identify these people and bring them in for surveillance, surgery and medical intervention. Now, this is a family that came into my clinic 3 years ago, just before I came to speak in this room for the first time, when UNESCO became a partner of the Human Variome project. The lady at the top had microsatellite unstable cancer. She had a mutation in her MLH1 gene, and I was told this was a variant of uncertain significance by our laboratory. My problem was that the lady in the white coat on your left is the one there who did not have that spelling mistake. The good news was she had not inherited it. The bad news was I didn't know whether that was the cause of the problem, because it was a variant of uncertain significance. This amino acid change had never been seen before, so no one knew whether it made a difference or not. Therefore, I couldn't act on it. This lady and her children faced 2-yearly colonoscopy and other regular medical checks because I couldn't be sure that they were not at risk. Fortunately, sitting just over there next to Rolf Simons, and Robert Hofstra, I was able to ask them. I said, have you seen this? They said, oh yeah, we've been to do some work on that. In fact, we're pretty sure that it is a pathological variant, because it affects the protein expression and it doesn't interact properly. And although our computer programs were uncertain, at least one of them said that it was significant, and indeed it had been reported in the literature subsequent to my colleagues actually doing the analysis on the first patient. So I was able to go back and say to this family, I have good news. I am now convinced that that spelling mistake did cause your mother's cancer. Therefore, I am now confident that you are not at increased risk of cancer, and therefore I do not need to give you 2 yearly colonoscopies. So you can see the powerful impact of that shared knowledge. And this is a slide from Dick Cotton's collection that he was shown in a recent lecture from our Insight Committee, where these are all laboratories doing different analyses of particular mutations, this particular mutation in the MLH1 gene. Some of them think it's pathogenic, some of them think probably not, depending on which technique you use to do the analysis. And so we have an international committee that gets together, converses on these topics, looks at this data, and then decides whether to put it into our database as a pathogenic variant or not. That is a hugely valuable contribution to the world literature. And it means that whether you're a public laboratory or a private laboratory, you can now go to the Insight database, look at that spelling change, and counsel families and give them the benefit of that shared knowledge. It makes obvious sense that we should do this. The Insight team derives its name from the International Society for Gastrointestinal Hereditary Tumours, which actually had its first meeting, combined meeting in Newcastle in 2005, and Finn now is its General Secretary. And we brought together in this little group here in Newcastle outside our centre all of the people who were running databases of the mismatch repair genes. Because there was one in Holland, there was one in Newfoundland, there was one in Finland, there was a bit in Australia. Everyone was doing their own thing. And we simply said, why don't we all get together and do one thing? And we'll call it the Insight database. And so, in fact, this is the model system for the locus-specific databases of the Human Variome project. And our plan is that we will have expert groups like this with their own special interest, looking at each family of genes or even individual genes, and then sharing that knowledge with the rest of us. And so some achievements. The Insight database now has 12,500 variants in the 4 genes, and we get 20,000 hits per month. So you can imagine the impact that's having across the world of people who are trying to actually counsel families like the one that I've shown you. And one of the problems, of course, is when you try and set up these databases, it's not always easy. Sometimes people who devoted their entire lives to collecting this data together can be a little precious, and they don't like to give it away. And so they have to be persuaded. And one of our tasks is to actually persuade our colleagues to pool their knowledge, because we are better together than apart. And in fact, one of the exercises that we undertook, and there's Dick at the very back there on the right-hand side, was to bring together a lot of opinion formers from around the world to actually get them to agree that we could collaboratively curate the Variome, as we put it. And in the front there, a very interesting character, Buck Strom from the States, who is actually the head geneticist at Quest Laboratories, one of the huge private laboratories in the States. We hear a lot about Myriad hanging onto their data, but Quest have donated their data to the system. They can see the benefit of this. If you do this for a living, the faster you can issue a report, the more money you make. So you don't have to be altruistic. This makes good commercial sense as well. I'd like to give you one other nice example of the challenge that we face. If you were to have gone back 500 generations in my family, I would have not shown great interest in my mother's breast milk at the age of a year, because it would upset my tummy, because I would have not had the ability to drink milk. We used to have a condition called hereditary lactose intolerance, because white people write textbooks and everybody else in the world couldn't drink milk. So we said that they had this genetic problem called lactose intolerance. So someone said, actually, there's more of them than there are of us. So we changed the name to hereditary persistence of intestinal lactase. We have the problem, or the variation, on the normal human trait, which is that once you've finished your breast milk, you should stop breastfeeding so the next baby can have the breast milk. And then about 7,000 years ago, Europeans, or about 7,000 BC, Europeans acquired this strange trait that we stopped switching off our lactase so we could carry on drinking milk all through our lives. And of course that ran hand in glove with the dairy industry and with our migration across Europe, and proved a very advantageous trait. You could feed without eating the cow, you could just drink its milk, a clear advantage, and the cow could walk with you to the next eating place. So basically this was a useful advantage. Which came first, the dairy industry or the gene? You can choose for yourself. But it was interesting that our Finnish colleagues a few years ago looked at this trait, and they found that there was nothing different about the lactase gene in white people compared to black people or Chinese people or whatever. The only difference they could find was a single letter change, 13,000 or so bases upstream, a single C to T change. And they got that published, and we all thought, whoa, that's not very impressive. But if they say that's what it is, that must be what it is. Now, then a very interesting paper came out in 2007, where the team went to Africa and they found 3 other tribes, marginally less successful than the Europeans, who had actually lived in Kenya, in Tanzania and Sudan, who were also pastoralists. They could also drink milk as adults. And they looked at their genes, fully expecting them to be our distant relatives, but surprisingly they weren't. They all had a spelling difference, and it was all very close to the last one, but it wasn't the same. The letters are along the bottom there, the C, G, T and G, were all in exactly the same position in the middle of another gene, 13,000 bases upstream. And when they took that little piece of DNA and stuck it into a cell in a laboratory, it turned out to be the on-off switch for lactase. So what had happened here was that 4 times at least, in fact at least 5 times in history, a mutation has hit the on-off switch for lactase, giving a population the ability to drink milk, and therefore changing their behaviour pattern. Now you can imagine how challenging that would have been to find if we didn't have the very easy phenotype that people can or cannot drink milk. We now know that there are literally thousands of non-coding RNAs in between the 20,000 genes that are like the fingers on the strings of the violin, controlling the expression of our genes. There are promoters, there are suppressors, there are all sorts of mechanistic differences between the genes that we have to understand if we are truly to understand disease. And that's before we even begin to think about how genes interact. My height, for example, is probably determined by 600 different genes, all interacting with different spelling changes in them. Added to which, whether or not I was breastfed or not, changes the methylation or chemical treatment of my DNA and also affects my growth. If my mother had been badly fed in pregnancy, that would have made me smaller. So there's a huge amount to learn, and we simply cannot do it unless we do it together. This is a big problem. This is a stone elephant from India. You will hear all sorts of new organisations coming along. The latest is the Global Alliance, to which Human Variome Project is a founder member and signatory. And the Global Alliance, I hope, is the beginning of a major upsurge across the world of investment in this field. But I don't think this is a replacement for the Human Variome Project. It's an enhancement of the brilliant concept that Dick Cotton gave us. So just a few more final achievements. The LOVD installations now contain 520,000 plus variants in 162,000 individuals. So when, as I met last week, a hospital lead who said, our hospital doesn't think it's ethical to share this data, and we don't think we're allowed to. Well, I know 162,000 people who disagree. I think we have to face this down. The only way we can collectively solve this problem is if we, in a secure and anonymous way, share this knowledge with each other. These are just the views. You can read the numbers. These are very impressive numbers. So when people say, what have the Romans ever done for us? What has the Human Variome Project ever done? Well, it's done a lot. It's only a beginning, but it's done the right things, and it's building on that. We've got colleagues all over the world becoming involved. The idea is, and you have to have a foothold in each country. The ethical, cultural, legal structure of every society is different. You cannot generalize how you will handle this information. And this is particularly true of things like we heard from America, where they are going to pass on information about people's unsolicited findings, whether or not they're at risk of high penetrance diseases. We in Europe think that's unwise, but we have a different healthcare system and a different legal system, and we can get away with withholding it. So, ladies and gentlemen, this is our picture from last year. The Human Variome Project is very much alive and kicking. It is, I think, going to go from strength to strength, and with the support already of the Chinese government, and I hope of British, European, American, Australian, the rest of the world, we will go from strength to strength and really begin to solve this problem. So in summary, sequencing will soon cease to impress. It is not the problem. The problem is analysing the data. Our ability to intervene will drive this process. People need to know. Angelina Jolie has done us a great service in that regard. We have a responsibility to integrate, and the UNESCO is perfectly placed to facilitate that. And we should never lose sight of the fact that failure to share this knowledge kills people. Thank you for your attention.